So now we're going to talk about different types of diffusion. Facilitated diffusion is still a form of passive transport. I'm not using energy. However, it's molecules that just don't go through the lipid bilayer. Okay. It can use transport proteins. It aids in many polar molecules and ions going across the bilayer. Pores are always open, okay? They just are like an open door. Gated channels open and close in response to stimuli. There are certain molecules we don't want going in and out of our cell unless we're being told to. Some molecules we want to have free reign, okay? Pores, those are the ones that have the VIP pass. Gated channels are the general admission ticket holders. Transport proteins share properties with enzymes. They are specific. They can be saturated, meaning that if I have too much solute, it only goes at a certain rate. I can only speed it up so much. And then they can be inhibited by something that resembles the solute, so they can be blocked. Active transport, these substances are transported against the concentration gradient. Okay, it uses, again, carrier proteins that are embedded in the membrane, but this uses ATP. This I need to use energy for. You have to think of active transport like paddling upstream in a canoe. If you want to actually go upstream, you've got to put energy into it. This, again, is just a nice example. It's just a pretty picture of how our conformation changes as one molecule goes out and the other molecule goes in, and it has a great deal to do with this phosphate group here that is being added and detached. Again, the sodium potassium pump, I'm not going to go over in excruciating detail with you guys. You guys should know that it is the example of active transport that we talk about. Okay, but I'm not going to ask you excruciating questions on the test on it. So vesicles can move substances. Sometimes we have substances that are too large for carrier proteins, big polysaccharides. And so we can have vesicles that do it. We have endocytosis, which means moving into the cell, and exocytosis, which means moving out of the cell, because in my universe where everybody takes Latin, endo means in and exo means out. Okay, phagocytosis is solid particles. We call it this because this is what phagocytes are, white blood cells, this is what they do in order to engulf a bacterium. Pinocytosis is fluid droplets. Remember I told you Pinot Noir, Pinot Grigio, Pinot Gris, they're all types of wine. Pinocytosis is cell drinking. This is just a nice little table that sort of goes through and lets you know. It is cool because these cell membranes will simply pinch off and fuse, which is nifty. Receptor-mediated endocytosis, this means that I only have this happen when a particular molecule binds with receptors and then it comes in, okay? This is special clearance. You've got to have high security passage in order to get in sort of situation. This is a retinal scan in order to get into the cell. Okay, so it's very specific. And this is how we control when we have hormones traveling through our bloodstream. They go everywhere, but they only affect certain cells. That has a lot to do with our receptor-mediated endocytosis. These are my three kinds. This is phagocytosis. This is eating a large solid particle. Pinocytosis, you can see the little things that are coming in here. It's sort of drinking it. And then here you can actually see the receptors that are embedded in the membrane. Receptor proteins bind a signal molecule, allowing the cell to respond to it. Some are attached to ion channels. This allows then certain ions to be able to travel through. Others cause a secondary messenger to come in, okay? So some, I now open the door. Others, I don't need the door open. I just need to get the information to the inside, okay? And that is that, ladies and gentlemen, and hopefully that will help.